Hello, dear students. In this video, we're going to talk about our new lesson. We're going to talk about transgenesis and the process of transcription and translation. What is transgenesis? Transgenesis is the process by which a gene is transferred from one species to another, and this gene is expressed in the later. For example, we have a gene that's found in human and it's not found in, let's say, in mice. So this gene will be transferred from human to mice, and this gene will be expressed in the mice. This process is called transgenesis. So let's repeat this. Transgenesis is the process where a gene is transferred from one species to another, and this gene is expressed in the other species. What the meaning of transgenic? Transgenic, it's an organism that carries and expresses a gene from another species. Let's say we said that transgenesis is the process where a gene is transferred from one species to another and expressed in the other species. So the mice, let's say we transfer a gene from human to mice. The mice, they carry the gene from human. So we call the mice, they are called transgenic mice. Why? Because they carry a gene that is from different species and they carry it. Okay? Let's have this experiment. In this experiment, we remove the egg of a mouse. We inject a human insulin gene in the form of DNA. So injection of a human insulin gene. So this gene is exclusive for human. So we inject it in the form of DNA in the male pronucleus, and then we transfer the number of alternate embryos that are produced into the uterus of a foster mother that's shown here. So the uterus of the foster mother, this is the foster mother. So we transfer the number of altered embryos into the uterus of the foster mother. So let's repeat what happened. We remove the egg from a mouse, and then we inject the human insulin gene in the form of DNA in the male pronucleus, and then we transfer the number of alternate embryo produced into the uterus of the foster mother. We get the result of 3 mice. We have the birth of 3 mice. The first one has a positive result. Positive result means that it's transgenic. So it expresses the gene of human, which is the human insulin gene. Whereas we have 2 that represent negative results, where there is no insertion of the DNA fragment, meaning that there is no expression of the human insulin gene. So here we have 1 positive, 1 that expresses the human insulin gene in mice. So this one is called transgenic. Why? Because it expresses a gene from another species and it's found in its DNA genome, while we have 2 negative results where there is no insertion of DNA fragment coding for the expression of the human insulin gene. Okay? So let's describe this experiment. So the eggs are removed from a mouse, then the human insulin gene in the form of DNA is injected. See here, using the passive voice, are removed. DNA is injected into the male mouse pronucleus, which is then injected in the female pronucleus. I'm starting step by step. This is the describe. Describe, we don't have to add any additional information. So you have to tell the story as it's mentioned in the figure without any additional information and using the passive voice. So the eggs are removed from a mouse, then the human insulin gene in the form of DNA is injected into the male mouse pronucleus, which is then injected into the female pronucleus. Then a number of altered embryos, they are transferred into the uterus of a foster mouse mother. After the birth of three mice and searching for the human insulin gene in their blood, two mice they showed negative result, meaning that there is no insertion of DNA fragment, while one mouse shows a positive result, and this mouse is classified as transgenic 1. This is describe. So as you see here, describe, I didn't add any additional information. I mentioned them as they came in the figure without any additional information or without adding any acquired knowledge. This is describe. Okay, so you have to pay attention to this. You have to start step by step as in the experiment. You have to use the passive voice and you have to write them as they came in the figure without any additional information. Here, interpretation. Interpretation, it's analyze plus indication. Okay, so analysis plus indication. Analysis, this is the analysis. Indication, the one in violet, this is the indication. So after injection of male pronucleus of a mouse previously injected by the human insulin gene in the form of DNA into a female mouse pronucleus and transferring the resulting altered embryos into the foster mother, the result was the birth of two mice with negative result, meaning that they don't express the human insulin gene, and we have one mouse, that is transgenic, meaning that it has a positive result and expressing the human insulin gene. This indicates that the human insulin gene in the form of DNA in mice could code for the production. This indicates that the human insulin gene in the form of DNA could code, because it may not, so it could code for the production of human insulin hormone in the transgenic mice, which is the positive result. Conclusion, a gene that's made up of DNA may code for the production of specific proteins, like in this case the human insulin gene in the form of DNA codes for the production of human insulin in mice. That's why these mice, they are classified as transgenic, because they express a human insulin gene that is not found in mice, and it's expressed in mice later on after its injection in the form of DNA. So you have to take care about this note, that a gene that's made up of DNA can code for the production of specific protein, example about this human insulin gene, and you have to know that transgenic mice here is the one that expresses the human insulin gene after its injection in the form of DNA. Let's move to talk about chromosomes. Chromosomes at metaphase, all of you know that they are made up of two sister chromatids. They are easily distinguished and they are aligned at the equator of the cell, forming what's known by equatorial plate. The substance that makes up the chromosomes often is called the chromatin. Chromatin is divided into DNA composed of four nucleotides. Four nucleotides, they are called A, T, C, and G. A, T, C, and G, they are the nucleotides that form the DNA. So DNA is composed of 4 nucleotides, A, T, C, and G. All of you know that the DNA is double helix, is double stranded. So we have two stranded opposite to each other. So let's say this one of the stranded, we have these nucleotides A, T, G, C, A. This is the first strand. The second strand should be complementary for the first one. So A, the complementary for A, it's T, and the complementary of T, it's A. The complementary of G, it's C, and the complementary of C, it's G, and also the complementary of A, it's T. So you have to know that A and T, they are complementary. C and G, they are complementary, as well as G and C, T and A. So A and T nucleotides, they are complementary. So if we have in the DNA a nucleotide A in the first strand, the complementary for A, it's T, and the complementary for C, it's G. Every 3 nucleotides together, for example, in this case, ATG, they are 3 nucleotides. Either they are called triplet, or they have the other name which is called codon. So you have to take care about this. Every 3 nucleotides, they form what's known by a triplet, or they are called codon. I prefer to use the word codon, C-O-D-O-N, codon. C-O-D-O-N. So every 3 they code for a codon. So for example, if I told you the first codon, where's the first codon? This is the first codon, A, T, and G. This is the codon. So A, T, and G together they form the first codon. If I told you the first nucleotide, it's the A here. Okay, so you have to take care about this, and you have to pay attention to this. So as I said, DNA is composed of nucleotides A, T, C, G, and every one of these, A is called a nucleotide, T is called a nucleotide, G is called a nucleotide, C is called a nucleotide, and A is called a nucleotide. Every 3 together, for example, ATG, these 3 together, they are called codon, or they are called triplet. A and T, they are complementary. C and G, they are called complementary also. Proteins, they are composed of histones and non-histones. So we're going to work about DNA, about the codon, about the nucleotide ATC. In the process of transcription, DNA undergoes the process of transcription, where they gave mRNA, messenger RNA. And mRNA, by the process of translation, they give polypeptide, which is protein. Okay? So DNA by transcription, they give mRNA, and mRNA by translation, they give polypeptide. So here, what is transcription? Transcription is the process where the mRNA, messenger RNA, ribonucleic acid, is produced based on the given sequence of DNA. Mainly the process of transcription, it occurs in the nucleus. What is the fate of the produced mRNA? The produced mRNA undergoes mutation, and then they are transported from the nucleus to the cytoplasm, where DNA is going to give mRNA by the help of the action of 2 enzymes, helicase and RNA polymerase. We're going to talk about them in the coming slides. So transcription is the process where mRNA is produced on the given sequence of DNA. Let's consider the sequence of DNA molecule shown here. We have this sequence. We have GCA, TGC, ATC, TGC, CAT, and TG. This is called the non-template. Pay attention here to the names. This strand is given the non-template. Maybe they will give you the strand is called template. So what's the difference? Non-template is called the non-transcribed. Okay? And it's called the coding. While the second strand is called the template, the transcribed, the non-coding. Okay, did you pay attention to this? transcribed, template, TT, non-coding. Non-transcribed, non-template, it's the coding. Okay, so this strand, it's the non-template, meaning that it's the non-transcribed, it's the coding. Why the complementary strand, it's non-template, opposite is the template. Non-transcribed, the opposite is transcribed, and the non-template, the opposite is the template. So how can we draw? We write down the sequence of the complementary strand. I'm not going to solve the first questions. I'm going to solve according to my own. So first, write the complementary strand. What's the complementary of G? It's C. What's the complementary of C? It's a G. What's the complementary of T? It's it's T. So here you have to pay attention. You have to write them one under the other, and they should be organized well in order to have the fully organized and fully correct sequence. Okay. Second, we have T, the complementary is A. G, the complementary is C, and C, the complementary is G. Here we have A, it's T, A, and G. The complementary of T, it's A. The complementary of G, it's C. The complementary of C, it's a G. The complementary of C, it's a G. A, the complementary is T, T, the complementary is A, and then we have T, the complementary is A, and G, the complementary is C. This is the complementary strand. They said write down the sequence of the mRNA. mRNA sequence. It's similar to the template strand. It's similar, to the coding strand, the non-template. It's similar to the coding, so it's similar to the one in the blue, to this, but instead of T, it's going to be replaced by U. So it's the same strand, but instead of T, it's going to be replaced by U. So let's move here. The same sequence, non-template. We said the non-template is called the non-transcribed, it's the coding. So GCA, we put it GCA, the same. TGC, instead of T, we put U. So in the mRNA, T is replaced by U. Okay? And ATC, A is the same. T is going to be replaced by U. C. If we want to continue this, T is going to be UGC, CAU, and UG. This is in the mRNA, the T is replaced by U. Here we worked on the non-template, which is the non-transcribed, which is the coding. Okay? So the coding or the non-transcribed or the non-template, the mRNA sequence is identical, is the same. Instead of T is replaced by U. Some of you ask me, can we use the template, which is the transcribed, which is the non-coding? Yes, but we have to do the complementary. Look here. C, the complementary, it's G. G, the complementary, it's C. T, the complementary, is A. A, the complementary, is T. But in the mRNA, there is no T. It should be U. So here U. C, it's a G. G, it's C. T, the complementary, is A. A, the complementary, is T. But it should be replaced by U. And then G, the complementary, is C. And so on. So the third part, but how can you relate the complementary template strand to the non-template to the mRNA? Template is the transcribed, is the non-coding. So it's complementary, but instead of T, it's U. While the mRNA strand, it's the same as the non-template, but T is replaced by U. And this is the case here. Non-transcribed, non-template, the coding is the same, but T is replaced by U. To produce mRNA, helicase enzyme. Helicase unwind the DNA. It unwind the DNA of the gene coding for the protein. RNA polymerase will synthesize the complementary strand. For the transcribed strand, will have the synthesis of the complementary. So this is the function of RNA polymerase and the helicase enzymes. Translation, we said the first process, DNA will give mRNA by the process of transcription, while mRNA will give protein or polypeptide by the action of the process of translation. It's the process by which an RNA, ribonucleic acid, in specific mRNA, goes for production of proteins, Some of these proteins will remain in the cell, others are exposed. So given the following coding DNA strand, AAA, CCC, GGG, CAA, TCG, give the sequence of the transcribe. It's complementary, where AAA, complementary is CTT, GGG, CCC, GTT, AGC. Specify the sequence. We specify the sequence of mRNA. It's the same as the coding. Instead of T is replaced by U. And we have to give the sequence of amino acid based on the sequence, based on the genetic code. So here, the genetic code table, where AAA here will give for protein lysine, CCC, every codon, every three will give will give an amino acid. Okay, GGG, CAA, UCG. Sure, you don't have to study them. They are given in this table. So this table, for example, UAA. Okay, the first letter is U. Here, the first letter is U. Second letter is A. For example, UAA, UAG, and UGA, you have to study them. They are called stop codons. For example, the first letter is A, the second letter is C. The first letter is A is here. The second letter is C is here. So THR. Okay? And so on. So this is the genetic code table. We'll start with the first letter and the second letter. First letter is here. Second letter is in the row. And the third letter, it's okay. So you have to study only three. UAA, UAG, and UGA, they are stop codon. Whenever we reach this, we have to stop because it's called the stop codon. And we're going to talk about this in details in the next presentation. So be ready. Thank you. Bye bye.